We're really looking forward to our webinar today. Jamie Jonker with National Milk is our presenter, of course, and his title is going to be FDA's residue study, the history, the results, and the implications. And those of you, many of you I know, realize that that study was initiated a long time ago. It was long awaited, the results, and so Jamie will be telling us all about that. And of course, the sponsor for the webinar today is Zoetis, and we appreciate their support as well. Always want to acknowledge and thank our co-hosts on this, Mike Hutchins down at the University of Illinois, and Jim Baltz as well, his sidekick there in the Animal Science Department down there. And always good to work with you folks as well. And with that, Mike Hutchins, I'll let you go ahead and introduce our speaker, and we'll get underway.
Well, thank you very much, Steve. It's a real honor to introduce Jamie Jonker here. He is the Vice President of Sustainability and Scientific Affairs for the National Milk Producers Federation located in Arlington, Virginia. Jamie grew up on a 100-cow dairy farm in New York State, got his bachelor's degree from Cornell University, and along with a master's degree also in dairy nutrition from Cornell University, and a PhD at the University of Maryland. So he is certainly, prior to that, he was involved in the National Academy of Sciences, the U.S. Environmental Protection Agency, and in the U.S. House of Representatives Committee on Agriculture. He is currently serving on the Federation's Animal Health and Welfare Being Committee and Environmental Issues Task Force. So, Jamie, thanks for taking time out of your very busy schedule, and we'll turn the program over to you. Thanks, and welcome to the HORDS webinar.
Great, and thank you for the opportunity to be here today. I want to thank my co-host from HORDS Dairyman and the University of Illinois to talk about this timely topic, which is the FDA's residue study, which was finally released just last month. But before we get into that, I'm going to talk a little bit about the history, then I'm going to talk specifically about the results of the study, and then I'm going to talk about some implications. And particularly on the implications part, what I would say is that this is a developing process. So anything that I say today is kind of a beginning to what we think may be happening in the future on drug residue testing for the dairy industry. So with that, I'll first start off with the history of the FDA process. And I first want to give everyone kind of a level playing field to understand the types of residue regulations that are out there for milk residues. And there's really two types of residue levels that the dairy industry is subject to at the farm level. One is called a tolerance, and this is established in the Code of Federal Regulations, and the specific code is 21 CFR 556. And in there, a drug may have a very specific level for a tolerance for a residue, such as 10 parts per billion. When there is no tolerance that is specifically listed, it means that the tolerance is actually 0. In practice, what that means is any detectable amount of that drug in milk would be illegal. And I think that's important as we consider that testing technology has gotten more sensitive over the years. The other type of regulatory level that the dairy industry is subject to is called a safe level. And this has been established by the Food and Drug Administration's Center for Food Safety and Nutrition and their milk, specifically their milk safety branch. These are the folks that oversee the grade A milk program. And there's a number of drugs that are contained in that what's called Memorandum of Information 05-5 that have a safe level to which we test at. An example of this is penicillin, which has a safe level in that MI 05-5 of 5 parts per billion. And so we know that milk at the farm level is subject to residue screening. And this is straight out of the Grade A Pasteurized Milk Ordinance, the last revision in 2013. Industry shall screen all bulk milk pickup tankers regardless of final use for beta-lactam drugs. That is that every tanker in the U.S. is tested for beta-lactams prior to that milk going into the processing system. While that's the only drug that we that all milk must be tested for. There certainly is other testing that goes on in the industry for business-to-business purposes, export purposes, and milk quality purposes. But no other drugs are tested for all of the time, other than beta-lactams today. However, the FDA commissioner can require additional screening if a potential problem exists. And I think we've got a very excellent story to talk about with what we've achieved through that beta-lactam testing of our milk truck tankers. Here in this slide, and this is data from the National Milk Drug Residue Database, we see the percent positive tankers over the past 20 years. And with the exception of a little uptick between 1995 and 1996, what you see is an exciting trend line of reduction from well over 0.1% of the tankers testing positive for beta-lactams in 1995 and 1996, all the way down to last year, where it was 0.014% of the tankers. And to give you an idea of what that is, that's approximately 1 out of every 7,143 tanker loads of milk. So we've had a very great success story there with beta-lactam residues in our milk supply. The other thing that the pasteurized milk ordinance requires is it requires testing of pasteurized grade A milk products prior to marketing. Again, this is a requirement for beta-lactams, and if a residue is detected, that product cannot be marketed. And over the past 3 years for which data is available, with nearly 130,000 tests conducted on those retail packaged samples, there have been no beta-lactam residues. Again, giving confirmation that on the milk side, we're doing a very excellent job, particularly with the beta-lactam drugs. Now I'm going to shift just a little bit and talk about what are we seeing in terms of residues in our dairy beef cull cows. And I'm first going to go back to a U.S. Department of Agriculture Office of Inspector General report from March 2010. And this was a review of the Food Safety Inspection Service. This is the part of USDA that has veterinarians in all federally inspected slaughter plants. So it's a program that they implement to look at the National Residue Program specific to cattle. And I think an important part of this report is finding number 2 that states FSIS needs to strengthen oversight of the National Residue Program, especially at plants slaughtering dairy cows and bob veal. What this means is that they determined through this Office of Inspector General report that there are more issues with residues related to dairy cows and bob veal than in other classes of cattle. And in fact, this is borne out by the fact that dairy cows and bob veal account for somewhere between 80 and 90% of all tissue residues that the Food Safety Inspection Service finds within an individual year. And that's not just cattle, but that's all cattle classes, all swine classes, and all poultry classes. When we look at the reason for these tissue residue violations, we shift from the USDA and the Food Safety Inspection Service to the Food and Drug Administration's Center for Veterinary Medicine, which has oversight over the use of drugs in animals. And when they have conducted investigations specific to dairy cull cow tissues, they've identified these causes as the top reasons for a residue to occur. Poor identification of cattle such that the producer may inadvertently ship an animal at the wrong time. Treatment records not recorded as a written record, so that there's not good information to determine whether or not the cow has passed the withdrawal time for meat. Not following the manufacturer or veterinarian prescribed label directions for the appropriate withdrawal period. Or not following the veterinarian's recommendation when using any drug. And I think those last two get to the point of what we call extra label drug use. And this is a policy that is a result of a federal law that allows veterinarians to prescribe certain drugs in a manner that is different from what the labeled indications for that drug are. In that process, they also have to provide a withdrawal period for both the milk and meat if the drug is used in a dairy cow. It's important to remember that in an extra label drug use application, a veterinarian must prescribe that medication. So if a producer is using a medication that's labeled for use in a dairy cow but does anything that's different from the label, such as they give it for a longer period of time, or you give a higher dose, or you give it in a administer it in a different way, such as IV versus subcutaneous, that's an extra label use, and that actually requires a veterinary prescription. And then finally, I think an important part here in these FDA investigations, and this is specific to those farms which have had a dairy cow tissue residue violation that have gone through this FDA investigation process, 70% of those farms do not have a valid veterinary client-patient relationship. And I think that's very important because that's actually quite different than what we see in the National Dairy Farm Program's animal care program, where across the industry we see in excess of 80% of farms having a valid veterinary client-patient relationship. So I think what we see here is potentially an area where the lack of a veterinarian working with the producer to make informed decisions could result in a tissue residue occurring. So what have tissue residues looked like in dairy cull cows? And here I have data over the past 10 years, and this data comes from the Food and Drug Administration and from the USDA. I do note that there is currently no available data for 2013. But we have an interesting process happening here. We had a very good educational effort within the industry starting shortly after 2007 when we saw a peak of over 1,000 dairy cull cows having a tissue residue violation. And we saw a very nice trend of those decreasing to a little under 400 in 2012. But for a variety of issues, we've seen an increase since then. And essentially, when we look over this entire decade process, there's virtually no difference between 2005 and 2014 in the number of tissue residue violations. I think this suggests that while we had success in our educational outreach starting about 7 years ago, perhaps we need to focus more again on that re-education process and reverse this trend that we've seen from 2012 to 2014. And so what types of drugs are we actually seeing in our cull cows? And this data from which is put into the Milk and Dairy Beef Drug Residue Prevention Manual that National Milk produces as part of the National Dairy Farm Program, shows that diceptiofur is the top residue within our dairy cull cow tissue violators. And this is a change from just 5 years ago when penicillin would have been our top drug. Penicillin is still the #2 drug, but there's been a change in the types of residues that we're seeing. We also see, rounding out the top 5, flunixin, sulfadimethoxine, and sulfamethazine. Those 5 drugs themselves account for almost 87% of the drug residues that were found from April 2013 to March 2014. So now that I've kind of laid out a history of both what we've seen happen in the milk testing side and the great results that we've seen in the Betalactam testing program and what we've seen in the dairy cull cow tissue residue side, I'm now going to get into the survey that the Food and Drug Administration conducted. And they started out with a hypothesis. Their hypothesis was that the same practices associated with tissue residues in culled dairy cattle may also result in drug residues in milk. And through the voluntary cooperation of the dairy industry, the Food and Drug Administration collected 1,912 samples in 2012 for this survey. They had 2 different groups that they were targeting, that they collected samples from. The first was the target group. This was 953 dairy farms with a cull cow tissue residue violation between 2006 and 2009. The other group, what we call the non-target group, is 959 random samples from other dairy farms. Each of these samples from both groups were analyzed for 31 different drugs, most of which were antibiotics, although there was some anti-inflammatory and some other types of drugs in there, but mostly antibiotics. And here you see a full list of the drugs that they tested for out of these 1,912 samples. What I want to highlight is in yellow. Those are 9 of the top 9 drugs for which we see tissue residue violations occur. So penicillin, flumixin, sulfadimethoxine, gentamicin, sulfamethazine, oxytetracycline, neomycin, tilmycosin, and ampicillin are 9 of the top 10 drugs. If you remember back to that pie chart just a few slides ago, you will recall that deceptiofur is now the top drug, and that drug was not represented in the survey. So this leads us to the first poll question for today, which is, what did you expect the findings of the FDA residue survey to be? Would there be no residues found? Option number 2, a small number of residues found but no serious problem. Option number 3, tissue residue target group has more residues than the non-target group. Option number 4, the non-target group has more residues than the tissue residue target group. And then option number 5, many residues across both groups, and that being really a serious problem for the industry.
Well, very good, Jamie. We're off and running here. We're almost 50% of the votes in.
My goodness, fast voting. Mike, what did you like on this menu?
Well, you know, I kind of went with the second one, but I'll be honest with you, Jamie. I thought there might be a sixth choice of saying more than that we found. In other words, I was nervous. But anyway, I'd probably go with number 2.
I would have picked number 3, I guess, and so we'll see who is the smartest. And I can see what you took the popular vote there, Mike.
That's right.
I think we've got a good response. So go ahead.
Okay, let's share it, Jim and Jamie. Your comments on the survey.
Great. I think the people on the webinar here have been really keyed into what we might be expecting, and I think it would be a great story. This 3% that no residues found. Certainly our Betalactam testing program demonstrates that we're doing a great job. For those that, and it's the over half that said small number of residues found but no serious problem, I think they're recognizing that we do a great job in the residue prevention programs that we have in place for milk. But we're only testing beta-lactams on every tanker, and this survey looked at 31 drugs, so maybe we could expect to find a few but not many. The 31% that chose option 3, the tissue residue target group has more residues. That's clearly the hypothesis that the Food and Drug Administration put forth. So that certainly is a reasonable one to do as well. Just a small number picked the non-target group has more residues, which to me I think means that folks think that if there is a residue issue that's different between the two groups, that the group that's had a tissue residue is probably a higher likelihood to have more residues than the non-target group. And then the last one, many residues across both groups with serious or significant problems. I think this was kind of the what if we really have an issue out there, and the big concern that we're only testing for beta-lactams. We know that there's a variety of other drugs that are being utilized in the dairy industry, and we have the potential that we could have a lot of different residues showing up in the results. But now going back to the outcome, we'll revisit that hypothesis that FDA had. And that was the same practices associated with tissue residues in culled dairy cattle may also result in drug residues in milk. And so here's the actual results from the survey. 99.3% of the samples were negative for drug residues, meaning they did not have any violative drug residues whatsoever. That being said, there were 16 residues found in 15 milk samples. So that means one of the milk samples actually had 2 drugs in it, and there were 15 milk samples out of that 1,912 that did, in fact, have a residue in them. Looking back at the hypothesis, FDA applied a statistical approach to see if there were any differences between the 2 groups. And they determined that no statistical difference existed between the target group, those farms had had a previous tissue residue violation from 2006 to 2009, versus the other dairy farms. They did find a numeric difference in that there were more residues and more drug classes found on those farms with a previous tissue residue violation, and I'll get into that information in a little bit. But I think importantly, FDA had this hypothesis, do the same practices associated with tissue residues in culled dairy cattle result in drug residues in milk? And in that case, the answer is no. So we're not seeing a systemic issue with those farms that have a residue in their dairy cull cow, resulting in them having residues in their milk that they sell to cooperatives and processors. But that being said, there were 16 residues found in 15 samples. And on this slide here, there's a lot of information, and I'm actually going to be visiting this slide one more time in this presentation. But I first want to talk about the number of drugs that we found compared to the 2 groups, and then the specific classes of drugs. So first, you can see in that second column called confirmed residues in targeted group, there's a total of 12 residues that were found, and that was 12 residues in 11 samples. In the confirmed residues in the non-targeted group, there were 4 residues in 4 samples. So there we see a numeric difference, but statistically we need to remember that there's no significant difference between these 2 groups. The other thing I want to point out here are the different drugs that we're seeing. Some may have thought that we might see maybe tetracyclines or a variety of sulfa drugs, or maybe even some beta-lactams, because the samples themselves were actually bulk tank samples and not tanker truck samples. But the drugs that we actually saw were ciprofloxacin, and you'll see that there's an asterisk on that one. Ciprofloxacin is a metabolite of enrofloxacin, which is what the FDA test detects, and therefore they couldn't distinguish whether it was really ciprofloxacin or enrofloxacin. And that'll be important later on when I talk about what these drugs can and cannot be used for. 1 residue in sulfamethazine, 1 in tilmicosin, 2 in tolanthromycin, 6 for fluorphenicol in the target group, those farms that had a previous tissue residue violation, and 4 fluorphenicol in the non-target group, and then finally 1 in the gentamicin category. And I think an important part for all 6 of these drugs is that none of these have an approved use in lactating or dry dairy cows. This means that any use of these drugs in the lactating dairy animal would have required an extra label drug use prescription by a veterinarian. Now I want to point to you for the top 2, ciprofloxacin and enrofloxacin, and sulfamethazine. Both of those actually are prohibited from extra label drug use at all, meaning the use of these, even if they were in fact prescribed by a veterinarian for a lactating dairy cow, is illegal, versus the other 4 drugs, if they were in fact prescribed by a veterinarian, including a residue withdrawal for meat and milk, would be acceptable under the drug label, extra label drug use policies that the Food and Drug Administration has that are part of the AMDUCA law. But we should not actually see a residue if those farms were following a veterinarian prescription for the withdrawal times on these products. So what did FDA say about these results? Remembering that there was no difference, no statistical difference between the farms that had a tissue residue violation in the past and the other groups. I think that the FDA messaging was very clear and very helpful for the U.S. dairy industry. And these are just 3 quotes that were pulled out of a number of the documents that FDA put forth on the day that they released the data. The first being, more than 99% of the samples are free of drug residues of concern, underscoring the safety of the U.S. milk supply. And I think there again, the safety, they didn't come away from this process thinking that there is a food safety issue with the U.S. milk supply. Really, they're considering that they have a compliance issue on the use of certain drugs and the withdrawal times for them. The second important key message, these findings are evidence that the nation's milk supply safety system is effective in helping to prevent drug residues of concern in milk. So the program that we're operating under through the Grade A Pasteurized Milk Ordinance is showing its effectiveness in keeping our milk supply safe. Then finally, the third key message is that the FDA believes that these findings of concerning residues in just a few of the samples collected reflect a snapshot in time from a limited number of individual dairy farms and do not pose a health risk to consumers. Throughout their entire messaging, what you see here is, 1, that the process we have in place is working, 2, that these results do not indicate a health risk to our consumers. So we have results that are showing that there are some residues occurring, but they're not occurring at a rate that causes an issue for potential health risk to consumers. So our milk supply is safe, yet we still have some residues that do need to be addressed. When this report came out, National Milk did also put out a statement, and this is a quote from Jim Mulhern, the National Milk President and CEO. Again, echoing what we heard from FDA, this report proves that America's dairy farmers are delivering on our commitment to providing safe and wholesome milk to consumers while working closely with state and federal regulators to continually improve the safety of our products. So I think there, we had this great story from FDA talking about the safety of the milk supply at the end of this survey that the industry was able to echo, saying that our milk supply is safe, and here's another test point that demonstrates that. So now that we've gone through the results of the FDA survey, let's look at what are the potential implications of these results moving into the future. And again, with this, we can go back to the FDA response when they released the report. And there's a few key items that I pulled out here that I think will give you an idea of what's going to happen in the future in relation to both milk and dairy cull cow tissue residue violations. The first is working closely with state regulators to consider modifying testing to include collecting samples as necessary from milk tanks on farms when investigating illegal drug residues in tissues involving culled dairy cows. This means that in the future, when FDA's Center for Veterinary Medicine conducts an investigation on a dairy farm that has had a culled cow tissue residue violation, they may take a milk sample from the bulk tank. I think this is important, particularly for our dairy producers on this webinar, to understand, in that if they take a sample, they will subject it to very rigorous testing for probably all of the drugs that were previously listed in this webinar, but perhaps even more drugs. FDA is continuing to refine its methodologies for looking at residues in milk and in other products. And so if you have, as a dairy producer, a culled dairy cow with a tissue residue violation, there is a reasonable probability that FDA in the future, coming onto your farm, could take a milk tank sample as well. The second key response from FDA is to utilize data obtained from this survey to help develop FDA's risk ranking for drug residues in milk that will assist NCIMS in including testing from more diverse drug classes in milk as necessary. I think the key points there are in that this is part of the information that FDA is utilizing within its risk ranking for drug residues in milk that the National Conference on Interstate Milk Shipments asked FDA to conduct. So this information is being used with a broader set of information to determine, is there a need for additional testing for drugs in milk across a more diverse classification of drugs. Then finally, the third point in the FDA response is to continue to work collaboratively with state regulatory agency partners and the dairy industry to strengthen the NCIMS drug residue testing program for grade A milk and to educate dairy producers on best practices. Again, this is going back to that state and federal regulatory process of the grade A pasteurized milk program, and using this information to determine whether or not there needs to be changes to the testing program that we do now, which is requiring all tanker trucks to be tested for beta-lactam drugs. So potentially the addition of other classes of drugs could be added. And again, talking about that education process on best practices for reducing residues both in milk and in our dairy cull cows. Now, just a few statements about the National Conference on Interstate Milk Shipments. The National Conference on Interstate Milk Shipments is the oversight body that puts together the requirements for the Grade A Pasteurized Milk Program. The Grade A Pasteurized Milk Program is a cooperative federal-state program to assess the safety of Grade A milk produced on the farm and processed through our Grade A plants. This conference meets every other year to consider changes to the program, and we are on the cusp of the next conference, which is being held April 24th through 29th of this year in Portland, Oregon. At this conference, the report findings will be used by FDA to consider changes in the milk residue testing system. There is a variety of proposals under consideration at this conference coming up later this week to consider changing how and what we test for residues in our bulk tanks and our bulk tanker trucks. I don't want to speculate as to what that might look like in the end, but I think what I can say is stay tuned. Those results will be available in a little over a week. So there will be information on any potential changes coming out of this conference by the end of this month. With that, I thought I would play for you here a video of Dr. Karen Jordan. She is a veterinarian and dairy farmer from Siler City, North Carolina, and she did this little video to talk about the results and what they mean.
So our milk supply is safe. FDA has confirmed our milk supply is safe, but there is the need for continuing education efforts among our dairy farmers and our veterinarians that supply service to our dairy farmers, pharmaceutical companies. We all need to work together to make sure that we have a safe food supply.
I think that's a very nice statement there that Dr. Jordan put together for part of the U.S. dairy industry's reaction to the report. You know, it's talking about this collaborative effort of working together on education for antibiotic stewardship, and that's a process that's not just at the dairy farmer, but it's through the whole chain. It's the dairy farmer, the cooperatives and processors, our veterinarians, the pharmaceutical companies, and our state and federal regulators. And that leads us to our next poll, which is, what is the major cause of antibiotic residues found in milk from this FDA survey? The first one is FDA testing limits are too low or sensitive. The second is poor record keeping on the farm, and that the dairy farmer cannot accurately determine withdrawal time. The third is poor identification of cattle, such that maybe the farmer is shipping the wrong cow at the wrong time. The fourth is the manufacturer or vet label withdrawal times are not accurate, so maybe those withdrawal times aren't accurate. And then the final is not following the manufacturer or vet label directions, which would, in essence, be the producer using their own extra label drug use without the prescription of a veterinarian.
Well, Steve, we're off and running again.
Wow, those are some tough choices, Mike. I'm going
Yeah.
to think about this a little bit here.
Well, I'm voting for the one that poor identification of cattle. I mean, we see problems. We do feed efficiency studies, Steve, here, and one of the big problems we can never get is how many cows are in a pen, because the pen dynamics are moving, and that becomes a very important number when calculating milk yield per cow and dry matter intake per cow. So I'm going to go poor identification of cattle, or cattle getting shifted around. But where are you going, Steve?
Well, I think I'm going to go with poor records, Mike. We'll see how. There's just a lot of things that can happen out there in the barns about things getting written down. So we'll see what the others think. Well, Mike, we're certainly not in the majority with our suggestions. So let's have Jamie discuss the results and see what he has to say.
What do you think, Jamie?
Oh, certainly. Yeah, we have people voting for every one, but there's certainly a dominant one that people have voted for. Let's start with the first one. FDA testing limits are too low or sensitive. I think we need to remember back that there are set by law residue tolerances for most of these drugs. In some cases, when there's nothing that is established in the regulations themselves, it's actually 0. So I could see how people would think that our tests are just getting too sensitive because they can detect smaller and smaller amounts. But I think we need to remember at the same time, particularly for those drugs where there is a 0 tolerance, any level in there is a violation and is a violation of the Food, Drug, and Cosmetic Act. And so while we have to realize that testing sensitivity keeps getting lower and lower, any drug in there actually is illegal and a violation for those with a 0 tolerance. For those that have a specific tolerance or safe level, we're testing to those levels, and so if there's a violation, if there's more than that safe or tolerance level, that's still a violation. So I could see how people would be concerned that we're testing too low, but we have to remember, there's either a set level that you're above or below. If you're above, it's illegal. If you're below, you're fine. Or if the level is 0, then any amount that's detectable is illegal. Poor records cannot accurately determine the withdrawal time. I think that there is an issue here that does happen for some farms, and that was borne out by what the FDA investigations have found in the past, in that sometimes you're not keeping accurate records. So did we treat the cow? When did we last treat the cow? Was it 6 days ago, or was it 5 days ago, or 4 days ago? And if you have uncertainty around when the last treatment was, it's very hard to make an informed decision on when you call the cow that you've really met that withdrawal time. Poor identification of cattle, shipping the wrong cow. This was a little bit lower on here. I think that can be a case, but I suspect that you can kind of put the poor identification and poor records actually together as a problem that kind of works hand in hand. Manufacturer or vet label withdrawal times not accurate. I think here I'm going to kind of actually break this one out into 2 parts. I think we only had maybe 2 or 3 people that indicated this one. But the manufacturer label directions, including withdrawal time, go through a very rigorous approval process through the Food and Drug Administration. So I don't think that a manufacturer label would be inaccurate, because I think that there would be severe economic consequences to a manufacturer having misinformation on their label. And when you combine that with the rigorous process that they go through for approval, that's probably just not a possibility. When you get to a vet label with a prescription through the extra label drug use regulations, they're doing their best information to provide an accurate withdrawal time on these drugs, and we do know that some drugs have a very significant milk withdrawal times, for example. But I think what we're getting here is really leading into choice number 5, which is not following the manufacturer or the vet label directions. I think we know, dairy producers have experience with some drugs, knowing that if they use it a little more, a little longer, a little different route of administration from the manufacturer's label directions, that that means they're using it off-label, and that leads to changes in the withdrawal time. So you can no longer do the withdrawal time that's on the manufacturer's label. Then with the vet label directions, that could be a case where where we move back up into that number 4 answer, where the vets are using the best information that they have available. But if they're not following the vet label directions, again, they would be using a drug in an extra label manner, and that would be illegal. So back to this table here, all 6 of those drugs, none are approved for use in lactating or dry cows. So we know that these residues are not showing up because there's a problem with the label and directions. They can't be used in lactating animals at all. And 2 of these drugs, the ciprofloxacin, which is most likely enrofloxacin, and the sulfamethazine, those 2 drugs themselves cannot even be prescribed by a veterinarian for use in lactating or dry cows. So those 2 drugs, it's even illegal to prescribe the drug. The other 4 can be prescribed by a veterinarian, but that prescription for a lactating cow must include a withdrawal time. And I want to focus in on one of these, because one of these drugs has 10 of the 16 residues. And I think it's important for folks to think about, perhaps how that may be being used in the field. This is, we don't know how an individual farm within this study was using the drug, because all of the results were blinded, so we do not know which individual farms had these residues. But I think we can look at the fluorphenicol, and I think an important point here to mention is that we think the withdrawal time for use in a lactating dairy animal is very long. We don't have good information available to us as a non-veterinarian as to what that might look like, but I would suggest that this can be weeks and maybe over a month, and that if you're using fluorphenicol on your farm, you need to be very conscious that the withdrawal period for milk may be very long. The other thing that I want to mention about a variety of these drugs is what they are approved for in other dairy cattle and or beef cattle. You'll see there on the second column from the right the status of whether or not this drug is approved for dairy cattle 20 months of age or younger and or beef cattle. And you'll see that 5 of these drugs do have approvals in those classes of cattle. And it may be coincidental, or it may be because these are kind of pretty broad spectrum drugs, but many of these are approved for bovine respiratory disease, the bovine respiratory disease complexes. And so, again, I have to be cautious here about speculating precisely why these drugs are being used in lactating dairy cattle, but I think it's at least interesting that the majority of them have BRD indications for other classes of cattle, just not for use in lactating cattle. And so where do we go from here now that we know what's been found, and we know that these drugs aren't approved for use in lactating dairy cattle, and even that some are illegal to use through an extra label drug use prescription. I want to point folks to our National Dairy Farm Milk and Dairy Beef Residue Avoidance Manual. It's our producer manual of best practices. And our sponsors, Zoetis, IDEX Laboratories, Elanco, DSM, and Charm Sciences, that help us make this available freely on our website. nationaldairyfarm.com in both English and Spanish. This includes excellent information on how to avoid residues in both milk and dairy beef. This is something that we update on an annual basis, so there'll be a new version of this coming out this fall for 2016. I also want to talk a little bit about this veterinary client-patient relationship. As you saw in that slide very early in the presentation about those farms that have had a tissue residue violation and an investigation from the Food and Drug Administration, 70% of those farms did not have a veterinary client-patient relationship. In contrast to what we see in the National Dairy Farm Animal Care Program, where about 80% of farms do have that VCPR. Clearly, working with a veterinarian, I think, is a factor to help reduce residues, or conversely, not working with a veterinarian is a factor that can lead to residues in tissues and probably in milk as well. I think another important thing to point to for education, and this comes out of the National Dairy Farms Animal Care Program. It's one of our points within that program is the need for a herd health plan, and I think this is great language here that the producers, veterinarians, and academic experts that put together that manual for us say an effective herd health plan emphasizes prevention, rapid diagnosis, and quick decision-making on necessary treatment of all sick or injured dairy cattle on the farm. This is kind of your tool as a farmer for determining how and when you use antibiotics, including making sure that you are adhering to all withdrawal times. And then here are the elements that are actually contained within the National Farm Animal Care Program's herd health plan requirements. So again, that top one, that veterinary client-patient relationship. A second one is vaccination protocols. Effective vaccination protocols can help reduce sickness in animals and reduce the need for the use of antibiotics. Daily observations of animals for injury or disease, protocols for calf management, painful procedures, protocols for cattle that develop disease or are injured, and that can include such things as determining how and when you decide to use an antibiotic, and also include in that the type of record keeping you would do to assure that you would be following the appropriate withdrawal times for both meat and milk. Protocols for difficult calving, protocols again for the prevention, detection, and action on common diseases, fly control, non-ambulatory animal management, protocols for euthanasia. Maybe we need to be looking at a little more as an industry. Are we making timely decisions on euthanasia rather than attempting to play a miracle worker and try to save a cow by using antibiotics when maybe that just won't work? Protocols to ensure food safety, again, that's following the withdrawal times. And then training programs for your animal care workers for detecting disease and injury, including record keeping and actions to be taken.
With our daily life on dairy farms, we are constantly working so that we don't have to use antibiotics. Meaning we are working on nutrition, we're working on ventilation and housing, and on vaccine programs. So once we have done those things to try to prevent disease, the antibiotic is the last thing in our toolbox that we reach for.
And I think Dr. Karen Jordan there, again, she's a dairy farmer and a veterinarian. She's also chair of the National Milk Animal Health and Wellbeing Committee. I think she sums it up very well. Antibiotics really, that's the last thing that you as producers want to use. And we need to have it in the toolbox, but we need to make sure we're doing a good job in deciding to use that tool. And with that, we've reached the end of the presentation, and I believe we now have time to open it up for questions.
Yes, we do.
Okay.
Turn it back to Steve. Steve, do you want to wrap up at this point?
Yeah.
And Jamie, do you want to click ahead one more PowerPoint? And I think you have one more. Very good.
Okay, Jamie, well, thank you very much for a very good presentation. Lots of good information, well presented. You really covered a lot of ground, and we had a very nice audience out there, and they've stayed with us all the way through, and we appreciate their participation. We thank you for the presentation, and of course Zoetis for their support of today's webinar. As always, we also want to thank Mike Hutchins, our co-host down there at the University of Illinois, and the two behind-the-scenes people, Jim Baltz, also down there at the University of Illinois, and also Patty Hurchin from Horge Dairyman up here, who make sure that things work for these webinars each time. Just another reminder that this webinar in a few days and all of the previous webinars are going to be available on our archives, horgedairyman.com webinars, and we've now reached a total of more than 52,000 archive views since we started our series. And as I think I mentioned to some of you earlier, 40 to 50 people a day go to our archives to view past webinars, and we certainly appreciate that interest. Also, here in a few days, those of you that were with us today will be receiving a very short survey about today's webinar. We appreciate your taking a few moments to respond to that survey to help us keep our webinar program on target. And as you can see on the screens, coming up next month, May 11th, Tom Jenkins from Clemson University is going to be talking about avoiding that summer slump in milk components. With today's milk prices, the components are very important, of course, and so we look forward to that. And also looking at another month ahead, June 8th, Sandra Gooden, University of Minnesota, talking about caring for calves in groups, and lots of interest in that these days, so we're really looking forward to that as well. With those comments, Mike, I believe we have at least one question so far. I'll turn it back to you.
Yeah, we have several actually. Steve,
Okay.
Jamie, can you click one more? And I think we have one more PowerPoint we'll bring up if you can. And we certainly, as Steve already has indicated, without sponsorship, these webinars couldn't happen. Here we go, Jamie. Hang on to your hats. Here comes a speed round. Is there any concern about extra label use in the future with veterinarians? Will we lose this tool because of some of the potential risks that came up in your discussion?
I think that we have to, as an industry, think about what does it mean with these types of results. Most of these drugs, those 6 drugs that we have there, actually already require a prescription to be able to pick up, meaning that most of them you can't just buy over the counter from your local farm and feed store. So I think that there is a potential that the extra label drug use regulations could change in the future. There certainly is a lot of interest within Congress on this and from advocacy groups in general about how we use antibiotics in food-producing animals. And results like this, do indicate a potential concern about how that extra drug use regulations are currently working.
Another question, Jamie, and that is, of the 16 samples that were picked up, were all of them actually zeroed? In other words, they're not recommended for dairy, so therefore they would be in that tolerance concern that you mentioned early in the webinar. Is that correct, or do you want to comment on that?
Yes, I believe that all of those have a zero tolerance within milk, which means that as tests get more and more sensitive, they can detect lower and lower amounts. But I'll fall back on any amount that shows up in the milk is illegal because zero means zero. And while we can't actually test to zero, we get closer to it as our testing gets more sensitive.
Perhaps a thorny question, and that is some milk is labeled as being antibiotic free, which some people argue implies that the others have antibiotics in. Could not all milk in the United States be labeled as antibiotic free based on the data you shared with us on product?
Oh, that's a sticky issue, and we don't like, many of us don't like seeing differentiation in the marketplace where there's not one. I'll leave it back to those that are responsible in the marketing of those products to assure that they are making truthful claims. And I think that's where I want to set that one is. Any claim that's being made on that label for a dairy product, whether it's antibiotics or some other sort of production practice or claim, that they have to be truthful in that.
Very good. What the other question was the 31 products or drugs that were selected. Were there any swine or beef? I mean, there were beef, swine or poultry included. Who picked the 31, or how were they selected?
So the drugs were selected by the Food and Drug Administration, and part of that process, they had a substantial outreach to the industry, to state regulators, the industry being both all the way from farm through processing, allied industry, and other concerned interests about the types of drugs to be testing for. And so that resulted in both what FDA's capabilities were, along with the types of drugs to look at, was a combination of all of that. So that's how those 31 drugs came about.
Very good. A little crystal balling. The interstate milk shippers will be looking again at this 400,000 somatic cell count. U.S. is unusual. We're at 750,000, where the world is at 400,000. Do you think this data will have some impact on maybe moving us down closer to the international standard of 400,000 somatic cell? Any ideas, comments, or thoughts?
Yeah, I have a couple of comments on that. One, we've tried, National Milk has had proposals the last two conferences to move to 400,000, and 4 years ago it was defeated by 1 vote. 2 years ago it was defeated by about 10 votes. So I think that I get the sense that there's not an appetite from our state regulators to move to 400,000. That being said, broadly across the industry, our industry average is right around 200,000 when we look across all milk produced in the U.S., which demonstrates the industry is really there. There certainly are farms that are not below 400,000, but broadly speaking, the industry is there. And we have a program that is being implemented through the USDA AMS branch. Their dairy branch there that is working to meet our requirements in the international marketplace. So it would be nice if it did go through this conference, and there is another proposal again this year, but it seems like there's not an appetite by our state regulators to go there, particularly with showing how well the USDA program is working. That being said, if there's any state regulators on the webinar here, certainly I think industry supports it, so make the right decision.
Okay, one more quick question. We actually have 2 left. Well, have you heard of any groups using this a little different direction? In other words, saying, well, there were residues in milk, and some of these could be action groups out there. Have you heard any kickback on the other side, where basically National Milk and FDA have said no harm, no foul? And have you heard anything from other groups?
So we were very concerned, I think the whole industry was very concerned as to how advocacy groups might use this information. And I think that FDA being so emphatic and so clear about the safety of the milk supply really took that concern and made it very small. We know that there is a variety of these advocacy groups that are looking at how drugs are used not only in dairy but across all livestock production. And I expect that at some point in time those groups will use results of this survey and other results from other livestock sectors to try and paint a bad picture. But the public use of that has been almost zero, and I think that's because FDA was so emphatic about the results, meaning that we have a safe milk supply, and that really was a great message for them to confirm so that we could say, as well as an industry, in echoing the safety of the milk supply.
Last question. You mentioned, I think, part of the answer here. Cipro and sulfa are approved for use in younger cattle and other species. For what purpose would that perhaps pop up on a dairy farm and maybe be a reason why the residue may have been there? Okay, so
enrofloxacin is a drug called Baytril 100 and Enroflox 100. And they're approved for use in cattle, beef cattle, with a 28-day withdrawal period for slaughter, and they're both approved for bovine respiratory disease. And that gets back to the discussion I had that most of these drugs are approved for BRD. It's a little hard to speculate that they're being used for BRD in lactating animals, but let's chalk that up as a possibility. I think the other thing is, these are pretty broad spectrum drugs, so they could be used for other purposes. And the sulfamethazine, again, is approved for use in, this one is approved for use in non-lactating dairy cows, 20 months of age or younger, and approved for use in beef cattle. And they are for, again, bovine respiratory disease. They also have approvals for foot rot. So there's a couple of other things that they're approved use for, but again, I come back to it's an interesting coincidence that most of these are for BRD, and again, not knowing exactly what happened on each individual farm, it's a little speculation, but there's either something with BRD, or because these drugs are broad spectrum, and that's one of the reasons why they're used for BRD, that they are efficacious for other reasons.
One more snuck in under the wire. This is the last one, I promise, because our time is gone. Any move, do you think, to revoke veterinary licenses if they can prove extra label use on lactating drugs when it was not approved for lactating cow use?
That is both a generally a state licensing issue with federal oversight on this, and I think if there are veterinarians that are prescribing drugs that's illegal for them to prescribe through extra label drug use, if they are caught, I would expect that there would be action taken on them. But I think that there's probably a combination of veterinarians prescribing drugs and maybe producers using drugs that they know have applicability in a situation. And so it's not real clear exactly how much blame goes to the veterinarian, how much goes to the producer. And that gets back to really the overall response that we need to do is we need to continue that education process from the producer and the veterinarian, the pharmaceutical companies, and our state and federal regulators to make sure that we're doing our best job possible to take the very small number of residues that we have, both in the milk and in the meat, and make them even smaller.
Well, Dr. Jamie Jonker, thank you very much for all your great answers and for your great webinar. Steve, closing comments.
Another thank you, Jamie, for a great presentation and a very nice job with the questions. We appreciate that. We appreciate Zoetis' support. Thank you, Mike, as always, for your co-hosting down there at the University of Illinois. And we look forward to having all of you out there with us on May 11th for our next webinar, Avoiding the Summer Slump in Milk Components. Tom Jenkins, Clemson University, will be the presenter. That's all for now from Fort Atkinson.